A very good morning, and we'd like to thank Josiane a lot for inviting me to come here and share some of our work with us in probably, I think nobody will doubt, the most beautiful city in the world. And to the organizing committee, and we had a wonderful session last evening on flavonoids and learning a lot of the chemistry of flavonoids. And I'm not a chemist, I'm a clinician scientist, so my work today will be more on looking at translational aspects of redox, particularly from the standpoint of mitochondrial signaling. I just had a quick comment to make here. Yesterday we had some comments on whether phytochemicals should be called phytochemicals or phytonutrients and etc. So it occurred to me that we have amino acids, right? We have essential amino acids and non-essential amino acids. So we could probably find a common ground and say that these are essential nutrients and perhaps non-essential nutrients rather than throwing them out of the window. Having said that, I think we'll get on to my talk here. So my lab actually for a number of years has been working on a variety of different stress signals and to ask a simple question in terms of cellular transformation, how these stress signals translate into oncogenesis and how some of these factors also result in chemoresistance. And so if you look at here, you have a plethora of different stress signals that a cell actually goes through or comes across during its lifetime. And most of the cells, the normal cells, mount an adaptive response here, which comes in terms of either differentiation or DNA repair if there is a DNA damage, growth arrest, and in conditions where the damage is irreparable, you execute the cell by either imploding through execution by suicide, or now we know that there are many other ways to kill a cell, and the cell ultimately dies. And all of this in normal cells is pretty nicely controlled by the presence of tumor suppressors. And we know the classical ones, B53, RB, APC, P10, and others. And so when these tumor suppressors function well, we don't have a problem. The cells go through these adaptive responses, and you come out with a nice-looking, healthy cell. But invariably, you see in tumor cells, in the clinical settings, a loss-of-function mutations of tumor suppressors together with oncogenic upregulation of tyrosine kinases and other signaling pathways. And that generates a very nasty-looking cell here, which we all refer to as a transformed cell. And so a lot have been said about transformation and how cells go through these processes. So our focus over the years has been looking at how redox signaling contributes to this process of carcinogenesis, not only from the standpoint of inducing tissue damage and death, but also from the standpoint of, like we heard yesterday in a very nice session on how ROS can signal, how these species are involved in growth and proliferative signals. And so over the next 20 minutes or so, I'll share with you some of our work, particularly one aspect of it that we have been doing on looking at superoxide and its relative derivatives and their ability to induce a resistant phenotype in human cancers and how this could be a very nice target for strategic therapeutic manipulation. So I don't have the time to go through this. This is all published work over the last 12 or 13 years using a variety of model systems, tumor cells, death ligation systems from the TNF receptor family, CD95 family, and others. We were able to actually show that the cells, in fact, do have a permissive ROS environment, which allows for the efficient execution of caspases and other proteases that are involved in the execution of the cell. And so if a cancer cell is in that environment, it's very sensitive to apoptotic stimuli. And if you increase this pro-oxidant state slightly or turn this oxidant state into a pro-oxidant milieu vis-a-vis superoxide radical, we were able to show that this pro-oxidant state can target a variety of signals in the cell, and that allows these cells to generate a resistant phenotype. And so the idea was then, could we play around with this model system and to see whether in clinical settings this sort of situation exists. And so we took this a little bit further and a few years ago proposed a very provocative model, which was that signaling by reactive oxygen species is a little bit tricky in terms of what species you're talking about. And we heard yesterday in terms of the redox potentials, how some of these ROS are very reactive and others are not. And so to cut the long story short, we were able to show that a tight balance exists between these two reactive oxygen species, and this could be altered by way of this very important superoxide dismutase and other enzymes that, as we all know, are radically changed in cells that are transformed. So when you keep these cells in this milieu, the cells actually undergo homeostatic growth, proliferative and apoptotic rates are kept in check. If you tilt this balance in favor of superoxide, and you could do that by clamping down superoxide dismutase or other systems, we were able to show that the cells actually enter into a proliferative phase, apoptosis is inhibited, and the predominant phenotype that you see is a resistant phenotype. And alternatively, if you tilt the balance in favor of hydrogen peroxide, you make these cells receptive to apoptotic stimuli. So this gave us a good model to test using some of the classical proteins that are involved in cell death and survival signaling. And so we simply asked whether this model could be superimposed on the two diverging functions of the BCL2 family. And as you all know, the BCL2 family members come in two different forms. There is an anti-apoptotic family, which is the classical BCL2 and BCLXL. These proteins are expressed on membranes like the mitochondria, nucleus, and the endoplasmic reticulum. And their function is to block mitochondrial-dependent death signaling. Alternatively, you have another set of proteins. These are structurally related proteins, but their functions are different. And these proteins belong to the pro-apoptotic family, BAX being the predominant family member. And their function is to target mitochondria for mitochondrial permeability induction or ER-induced changes that results in execution signals. So our question simply was, could it be possible that the survival effect of BCL2 family members is a function of a tilt in this balance towards superoxide, whereas hydrogen peroxide predominates when BAX and BAC and other pro-apoptotic members of the BCL2 family are engaged. And I don't have the time to go into this. This is already published. So we showed a number of years ago that yes, if you increase intracellular hydrogen peroxide concentration by exposing cells to chemotherapeutic drugs, or exogenous addition of hydrogen peroxide at low concentrations, not at necrotic concentrations, you could induce a conformational change in BAX, which is a pH-dependent effect. And this allows for BAX to translocate to the mitochondria, and this oligomerization of BAX induces further increase in mitochondrial-dependent ROS production. And this generates a positive feedback loop by which hydrogen peroxide then signals through mitochondria, induces mitochondrial lipid oxidation and other changes, and the mitochondria become permeabilized, cytochrome C is released, and apoptosis is induced. What I'm going to spend most of my time on is the other part of the equation, superoxide. And so I'm going to show you some model systems that we actually first used to address that issue. So this is a paper that we published a few years ago, and we used this model, this leukemia cell model, where we ligated the death receptor by this ligand, CD95 ligand. And the idea was to use these cells because these cells are inherently dependent on mitochondria for execution signals. So if you take away the mitochondria, these cells don't respond to apoptotic stimuli. So you could do that by just expressing BCL2 onto these cells, and once you express BCL2, these cells are not responsive to receptor ligation. And you can see here that these cells are quite resistant. So the question we simply asked was, is it possible that BCL2, in addition to functioning downstream of the mitochondria, has actually a function upstream of the mitochondria by generating superoxide or increasing superoxide radical and its derivatives, and knocking out the activity of the caspase. So to do that, we actually generated a model where we clamped down the expression of superoxide in these cells by either a dominant negative of RAC1 or by pharmacological inhibition of the NADPH oxidase complex. And what we were able to show was that, yes, if you do that, you actually don't need the mitochondria for the execution signal. You can drive the signals directly from caspase 8 to caspase 3. So basically what we were doing was switching a type 2 mitochondrial signal into a type 1 receptor-induced cell death signal. So the question simply was, how does BCL2 impact cellular redox status? Most people in literature would associate BCL2 with an antioxidant activity. And what we were able to show was that, in fact, BCL2 was able to engage this rate-limiting enzyme in the electron transport chain. So one of the studies that we did was to see whether any of these complexes was involved. And a very talented graduate student was able to figure out that this complex, this COX subunit here, which is about 13 protein subunit, 10 of them actually are imported from the nucleus, was somehow involved in the upregulation of superoxide production in the mitochondria upon overexpression of BCL2. And what he was able to show was that if you actually took these cells and you looked at the oxygen burn rate by simply looking at oxygen consumption in the mitochondria, you could see that the BCL2 overexpressing cells burnt oxygen at a much more efficient rate. And this could be also shown here with state 4 and state 3 respiration in the presence of a substrate. And most importantly was that if you measure the activity of COX here, this is not cyclooxygenase, by the way, this is cyclooxygenase, you could see here almost a 250 to 300% increase in the activity of this rate-limiting enzyme in cells that were overexpressing BCL2, both in leukemia as well as in a cervical cancer cell, HeLa. And so this is a busy slide, but the question simply we asked was whether there was a function of BCL2 somehow promoting the assembly of this complex in the mitochondria. And I just want to point your attention to these boxes here. So this is the COX V alpha subunit, which is imported from the nucleus into the mitochondria. And these are just fractions. And you can see here, when you overexpress BCL2, the recruitment of V alpha to the inner mitochondria is much more efficient and robust. And we also did blue native gels to show really that the complex was much better assembled in cells that were overexpressing BCL2. We could also show that there was somehow a shuttling of this complex from the nucleus to the mitochondria, which is shown here in BCL2 overexpressing cells. You can see here a lot more in the mitochondria than in the nucleus. Furthermore, Yong went ahead and asked whether a possibility existed that BCL2 physically interacted with a subunit in the cytochrome C oxidase complex. And we were able to show that, in fact, if you just did a conventional IP Western, that COX V alpha nicely co-precipitated with BCL2, whereas other family members like BCLXL, BAX, and BAC showed no activity towards V alpha subunit. So this suggested, and we went ahead and showed that this was not only in cell lines, but also in some clinical samples derived from patients, you could show that, in fact, this increase in V alpha in the mitochondria was higher in cells that overexpress BCL2. So we were still looking for a causative mechanism by which BCL2 was able to increase superoxide production. And one hint came when we actually did these experiments in the presence of a RAC dominant negative. So when we transfected these cells with a dominant negative of RAC1, and this is just measuring superoxide levels in these cells, you can see here when you silenced RAC1, you can completely bring down the level of superoxide it increased in the presence of BCL2. You could do the same by measuring mitochondrial superoxide production here with a RAC inhibitor. So this is a RAC inhibitor that blocks the activity of RAC. And you can see again here that the increase that you see with BCL2 is completely reversed in a dose-dependent manner with the RAC inhibitor. And RAC, in fact, was shown to co-precipitate with BCL2 in a variety of different cell lines. And you can see here RAC1 and BCL2 nicely co-precipitating in Jurkat, Raji, NCE, and BCL2 cells. And so this suggested to us that maybe there is a complex or a tripartite complex of RAC, BCL2, and perhaps COX V alpha that's playing a role in the ability of BCL2 to function as a pro-oxidant in these cells. We went ahead and asked which domains within BCL2 and RAC, if this interaction was possible, could be responsible for this interaction. And so we did a computer modeling here. So this is just the RAC and the effector domains of RAC. And this is BCL2. This is a very important non-structured loop region of BCL2. Most of the pro-apoptotic proteins of BCL2 bind to this BH3 groove here. So we actually were able to predict that this loop region might have a role to play in its ability to interact with RAC1. So why is this loop region important? This loop region is important because there are some very important phosphorylatable sites within these loop region, particularly 3 of them that I'll mention in a few minutes. And so what we were able to show was that, in fact, if you block this loop region by peptide loading, so these are cell-permeable peptides that block the BH3 domain, which is just next to the loop region, you can see here that you can completely block the co-localization of BCL2 and RAC. And you can show that also in the Western blot, that when you loaded these peptides in these cells, the BH3 inhibitor peptide, you could see completely the inhibition of RAC interaction with BCL2, suggesting that this region that is next to the BH3 domain could be critical in its interaction with BCL2. So the question simply was whether RAC, as we know, is a GTPase. Do you need RAC to be active to bind to BCL2? It has to be active in order for it to produce superoxide. And so we loaded these cells with GTP gamma or GDP, and you can see very nicely here that BCL2 interaction only takes place or predominantly takes place with RAC when RAC in its active form, in its GTP bound form. Plus, we were also able to show that this loop region, which contains 3 important residues, phosphorylatable residues, the S70, there are 3 important residues here, serine 70, threonine 69, and 87. And so we simply asked whether this could be a possible mechanism by which phosphorylation of BCL2 drives this interaction. We made a mutant of this BCL2 as serine 70 by just removing this serine to alanine, and you can see here that this falls out. If you make a phosphorylation mimetic mutant with a glutamine at the end, you can see robustly the increase in RAC interaction with BCL2, suggesting to us that S70 phosphorylation of BCL2 could be a critical player here. Now, why is S70 phosphorylation of BCL2 important? If you read the literature, you'll find that BCL2 is phosphorylated at multi-sites, and multi-site phosphorylation of BCL2 actually inhibits its anti-apoptotic activity. But you actually look at the monocyte phosphorylation of BCL2 at 70, BCL2 is stabilized and functions much more efficiently as an anti-apoptotic protein. And this is shown here. This is just an assay to look at cell survival. You can see here that when you mutate the S70 residue, the ability of BCL2 to protect cells from adipocyte is completely lost. A phosphorylation mimetic mutant nicely restores this activity. So suggesting to us that the S70 phosphorylation of BCL2 could be a critical target here. Now, what phosphorylates S70 or serine 70 of BCL2? So in the literature, you will find reference to ERK and JNK as kinases that are involved in the phosphorylation of BCL2. So the first question we asked was, does the increase in superoxide signal as an oncogenic signal by way of impacting the phosphorylation status of BCL2? To do that, we actually again took the same simple approach. Either inhibited superoxide dismutase pharmacologically, or we knocked out using 4 different gene sequences to target the expression of SOD1, which is shown here. So if you can see here nicely, only the S70 residue of BCL2 is phosphorylated in the presence of an inhibitor of SOD1. You don't have any effect on S87 or T69. You see the same effect when you use 4 different targeting sequences to SI-SOD1. As you can see here, SOD1 is silenced nicely, and you can see nicely concomitant phosphorylation of S70 BCL2. So linking very closely superoxide or its derivatives in this phosphorylation of BCL2. We were able to show that this function of phosphorylation induced by superoxide was not a function of kinase activation. We inhibited kinase, ERK or JNK or silenced them. We were not able to block this. So we reverted to look at whether this was an effect on the phosphatase. As you know, the phosphorylation status of a protein is a function of kinase activation and phosphatase that take away the phosphate group. And so we were interested in this phosphatase PP2A. As you know, PP2A is a very complex enzyme. You need these 2 subunits AC to then come together with this beta subunit, which brings the substrate and forms this hollow enzyme assembly. Now, PP2A enzyme assembly and the beta subunits, there are many, and it's a very redundant enzyme in itself. So we were able to isolate a beta subunit, a B56 subunit, that specifically targeted BCL2 for dephosphorylation. All this work that I'm showing is actually not published. So what we were able to show is that, in fact, if you did a pull-down assay, and this is basically pulling down PP2A, the AC subunits with BCL2, you increase superoxide in the cell, you completely block the interaction between these two substrate as well as BCL2, the holoenzyme. You do the same thing by silencing superoxide dismutase 1. You see again here PP2A interaction with BCL2 falls off. So we generated a model from this, and I don't have the data to show you that we have identified the residue within PP2A B56 delta subunit, which is amenable to nitrositive modification. So this is the model here. This is how BCL2 is dephosphorylated. Phosphorylated BCL2 at S70 sits at the mitochondria. PP2A comes along, nicely dephosphorylates it. Phosphorate goes away, and BCL2 loses its ability to function as an anti-apoptotic protein. What happens when you have an increase in superoxide concentration in the cell? As we heard yesterday very nicely, you got plenty of nitric oxide, produces peroxynitrite. Peroxynitrite targets this tyrosine residue. Now you have this beta B56 subunit hooked up with this modified tyrosine residue. The AC subunit tries to come and talk to it. It doesn't want to sit and sleep with it. So the end result is BCL2 stays phosphorylated at the mitochondria and functions as an anti-apoptotic protein. So these sets of experiments have actually given us a new paradigm, or probably suggesting a new paradigm, a new way of thinking in terms of the biology of the BCL2 family. We already know that the BCL2 family is very important. There are many mechanisms by which it regulates oncogenesis. And what we are able to show is that mitochondrial metabolism could be very important, generating a pro-oxidant state that then allows for cancer progression to take place by itself targeting the stability of BCL2 via its phosphorylation. And using that model, we are actually now looking at a variety of different pathways. We heard about epigenetics yesterday, looking at redox-sensitive genes in terms of proliferation, migration, and transcription. And we have some data that hopefully we'll present sometime on AKT and PI3 kinase in this regard. And obviously, we'd like to the most important slide has disappeared, which is, I'll take the questions in the meantime, just to thank a lot of my students and people who have done the work, which are right here. A lot of the initial work was done by a postdoc, Jay Shri. Iwan did the work and is continuing to do the work on the PP2A. Chiseong did the work on BCL2 and V-alpha, and Ratika and Kangjia were the ones involved with the RAC1 work, and our collaborators here and the funding agencies, and I'll take any question at this point. Thanks.
Thank you very much. Informative presentation. I think we have time for several questions, please. Yes?
Congratulations for a very nice talk.
Thank you.
On results. My question concerns PP2A, the role of PP2A on BCL2, because PP2A, especially the catalytic subunit, it's redox sensitive, has the thios in the active center. So the effect of superoxide production may also affect the catalytic subunit. Could it be involved in the phosphorylation of
yeah, that's a good question, although when we did these experiments, so if you look at the literature, there are two different subunits associated with BCL2 dephosphorylation, the B56 alpha subunit. We actually didn't find the B56 alpha, we found B56 delta. But these are redundant units, so they can do each other's work. What we were able to show actually was that when you do this, the holoenzyme activity doesn't go down, which suggests that it's not the substrate. So PPA2A is doing pretty well with other B56 subunits. However, we see the same effect with Tau, CDC25, and more recently with P53. So there may be a general inactivation of PP2A B56 subunits just by nitrositive modifications. So that we have to look at.
Thank you.
Good question, yes, please. That was
have any determination of the SOD activity? Because these changes in superoxide should have an impact in the biosynthesis of SOD.
So we have done when we do silencing of SOD, gene silencing, we do two things. One is to show that the SOD activity is clamped down. And secondly, to see that superoxide production is increased, or not production, but superoxide is not dismutated to hydrogen peroxide. So we measure the ratio with all the assays that are available right now. But silencing of superoxide dismutase definitely inhibits its activity. So we can show that we can almost 100% silence the expression of SOD1. So if that is your question in terms of whether the SOD activity or superoxide itself could impact superoxide dismutase activity. We do see some MNSOD activity, which may be a compensatory increase in MNSOD activity. But that's another story for another day.
This is another thing. This is the other compartment.
Exactly. Which is not in the cytosol mostly anyway. But that's another story that I can tell you maybe at some other time.